Hi everyone, this is Fahmida. Welcome to today's session on first week of embryonic development. In this session, we will know about what changes occur in the first week of embryonic development. For better understanding of this class, we have to have a clear conception regarding gametogenesis, ovulatory cycle, and uterine cycle. Events which take place in the first week of embryonic development are fertilization, cleavage, morula, blastocyst, and implantation. The next couple of minutes I will discuss about these steps. Fertilization. Fertilization means the fusion of two mature germ cells, an ovum and a spermatozoa, form a single cell called zygote. Now we will know about the site of fertilization and the preparation of the ovum and the spermatozoa just before fertilization. Fertilization occurs normally in the ampulla of the uterine tube. Now question, how the oocyte reaches to the site of fertilization? After ovulation, the secondary oocyte with a single polar body surrounded by the corona radiata is discharged into the pelvic cavity. Then the oocyte is carried into the uterine tube. After then, by the movement of the both femora and the uterine tube, the oocyte reaches to the site of fertilization. Now the second question, how the sperm reaches to the site of fertilization? The sperm reaches to the site of fertilization by the movement of their tail and the uterine cilia, and also by the chemoattraction. Before fertilize the oocyte, the sperm must undergo two processes. Number 1, capacitation. Number 2, acrosome reaction. Capacitation. Capacitation occurs when sperm reaches in the female genital tract. What happened there? The glycoprotein coat and the seminal plasma protein are removed from the plasma membrane that overlies the acrosomal region of the spermatozoa. The second one is acrosomal reaction. It occurs after capacitation, when the proteolytic enzymes are liberated from the acrosome in order to penetrate the zona pellucida. Let's see what are the barriers should have to cross sperm to reach the oocyte. The barrier protect the oocyte are from outside to inwards, corona radiata, zona pellucida, and vitelline membrane. We know that per ejaculation 200 to 300 million sperms are ejaculated. Among them, only 300 to 500 sperms are reaches to the ovum, but only one can penetrate the barriers and can unite with the oocyte. Fertilization process. Only sperm's head can penetrate the zona pellucida. Sperm's head and its nuclear envelope swollen and form male pronucleus. On the other hand, the nucleus of the mature ovum also swollen and form female pronucleus. These pronucleus are haploid, meaning they carry the half number of chromosomes of the somatic cell, which is 23 in number. Then the pronucleus are fused each other and form zygote. This zygote contains 23 pairs of chromosomes. Now we will know what are the effects of fertilization. Number 1, in ovum, completion of second meiotic division and formation of mature ovum with second polar body. Number 2, restoration of diploid number of chromosomes in the zygote. Number 3, determination of chromosomal sex. Number 4, determination of polarity of the embryo. Number 5, Initiation of cleavage division. Cleavage. It is the repeated mitotic divisions of the zygote, resulting into a rapid increase in the number of cells. During this process, the zygote becomes two-cell stage within 30 hours after fertilization, four-cell stage 40 to 50 hours after fertilization, eight-cell stage within 72 hours after fertilization. The zygote gradually migrates to the uterine cavity from the uterine tube. After 72 hours of fertilization, the zygote forms morula. Morula is 16-cell stage embryo. It has mulberry appearance. The zona pellucida is still present. The inner cell must form embryo proper, and the outer cell must form protective and nutritive covering of the embryo. The next stage is blastocyst. The blastocyst is formed within zona pellucida on 4th or 5th day of fertilization. Fluid penetrates through the zona pellucida into the intracellular space of the inner cell mass. Gradually, the intracellular space becomes confluent, and finally a single large cavity is formed, called blastocele. The zona pellucida disappears at the end of the 5th day of development. Here, the inner cell mass called embryoblast and the outer cell mass called trophoblast. Implantation is the final step of first week of embryonic development. Penetration of blastocyst into the functional layer of secretory phase of endometrium is called implantation. The normal site of implantation is posterior wall of fundus of uterine endometrium. Subsequent development takes place from here. This is all about the first week of embryonic development. Now we can summarize it. Summary of embryonic changes during first week of pregnancy. Fertilization takes place in the uterine tube. The zygote formation is the result of fertilization. The zygote divides continuously by cleavage to form a ball of cells called morula. The zona pellucida ruptures after due to increase in cell population. The cells organize into two layers, inner cell mass and outer cell mass. The inner cell mass are embryonic cells going to form embryo proper. The outer cell mass are trophic cells going to form the placenta. Implantation occurs. Penetration of blastocyst into the uterine mucosa. Thanks everyone for keeping up with me.